This is being recorded. I am Terry Conneran with KRAS Kickers and excited to see you guys here today. We are delighted that we have the opportunities to do partnerships with folks like Amgen, and their sponsorship has allowed us to be able to bring a speaker in to you today. Today we have with us Mark Awad from Dana-Farber Cancer Institute. He's the Associate Professor of Medicine at Harvard Medical School, medical oncologist, I almost spit it out wrong, Dana-Farber Cancer Institute. He's also a wonderful guy and a new dad, so congratulations, Mark. And welcome, we're really excited to have you here today and hear about the SPARK thing and everything that's going on in KRAS.
Thank you so much, Terry. It's always a pleasure speaking with you and speaking with the KRAS Kickers, and it's really the highlight of my week whenever I get to talk to patients and their loved ones, advocates, and it really helps to fuel our work and our research. So it's a real pleasure to be here. I'll try to monitor the chat as well to see as questions come in, but I always like when this is as interactive and informal as possible. So feel free to send questions through the chat or to interrupt and ask questions along the way. I thought we could talk a little bit about how we approach understanding drug resistance for KRAS. As Terry mentioned, I wanted to highlight a protocol that we just opened that everybody is invited to participate in, no matter where you are in the country, which I'll be able to describe in more detail. So Terry, thank you for the kind words and introduction. Shall I maybe share some slides and we can use that as a sort of launching point? Okay, that's great.
I think if your shares should come on, if it's not, there we go. Yep, you're good.
All right, terrific. Well, I thought we would try to highlight what we've been working on to understand resistance to KRAS inhibitors. And again, I want to make this as accessible as possible. So if something's not clear, then I haven't done a great job of explaining. So please just interrupt, and I'm happy to take any questions along the way. And Terry, also, if you see or others see questions coming in that I'm not monitoring, feel free to, as you know, just jump in and interrupt. So, why are we interested in studying drug resistance? Why focus on this in the first place? And how do cancers develop resistance? We've done a lot of work on KRAS G12C in particular, but as new drugs are coming into clinic and in clinical trials, hopefully this approach will be able to be applied to many different types of KRAS lung cancer. And what strategies can we take to try to overcome or hopefully even delay the emergence of resistance? And I want to talk about this SPARK protocol, which is a remote participation study to collect blood samples from patients who are experiencing resistance to KRAS inhibitors. And I'll get into some details as to why we're excited about this study. So I know this group is really familiar with KRAS, and I'll use this diagram to kind of highlight how we try to study resistance. So what is KRAS? So this is a diagram of a cell. The top lines here are the outer membrane of the cell, and then down here is the nucleus of the cell that houses all the DNA, and that's where DNA needs to divide to make new cells, which is a normal process for normal growth and development. When you're a baby and have to make many new cells, these are normal signaling processes. And so there is a normal step to tell cells that you need to grow and divide. And usually there's some kind of signal that's outside of the cells that's a growth signal, and it tells the cells, okay, now it's time to start growing and dividing. And one such example that I showed here is the epidermal growth factor, also called EGF. And this is a signal that can float around when necessary, and it binds to a receptor on the cell called the epidermal growth factor receptor. And that really tells the cell to start growing and dividing. But how do we transmit a signal from outside of the cell all the way down to the inside of the cell into the DNA or to the nucleus? And that's where KRAS and other proteins come in. So KRAS, or RAS in general, is a protein that transmits the signal from the outside of the cell through the cell surface all the way down to the inside of the cell here. And as I mentioned, usually this is a very well-controlled, regulated process. You only need to transmit this signal during normal growth and development. Therefore, the signal is really only turned on when a growth factor is present during normal development. But what are KRAS mutations? And so here I'm indicating by this red asterisk that there can be some abnormalities in the KRAS protein that leave this pathway, this signal on. So even when there's no growth factor present telling the cells to grow or divide, this signal gets stuck on even without any growth factor. And so it keeps the gas on the accelerator of these cells, and it tells the cells to continue growing and dividing even when this is inappropriate, when there's no growth factor. So we call these mutations activating or driver mutations because they drive the cell's growth forward. So an example, there are different types of mutations within KRAS, and many people are familiar with the notations, but there can be, for example, KRAS G12C or G12V or G12D, G13 mutations, and others. And what does that mean is a protein is made up of a string of different amino acids that are linked together. Each amino acid has a number along the string. And there's a normal amino acid that should be there. And then with the mutation, you switch it out for an abnormal amino acid. So G12C means at amino acid position number 12, what's normally a G, or what's called a glycine amino acid, is switched to a C, or a cysteine. Or a G12V means the normal G at the position is changed to a different abnormal amino acid called valine, and so on. And all of these mutations have the same net result generally, that it results in abnormal growth. And here I'm a thoracic oncologist, so I see mostly lung cancer, but this really does apply to all KRAS mutations across all different tumor types. So in lung cancer, and I'll talk about lung cancer because this is where we have our first approvals for KRAS, but again, we're learning more and more about this across different tumor types. But in lung cancer, in colorectal cancer, in many other tumor types, KRAS is one of the most commonly mutated genes. This is a pie chart of different mutations we can find in lung cancer, for example. And the biggest slice of the pie here is represented by KRAS. And there are KRAS G12C mutations and then other mutations as well. But this has been really important for us to start identifying because now we have these G12C inhibitors. So how do these inhibitors work? So here in this same diagram again, these are drugs that will block the mutant form of the KRAS to sort of turn off this downstream signal so that there's no further signal to the inside of the cell, to the DNA, to start dividing. And these drugs are very potent. Now we have our first approval with a KRAS inhibitor called sotorasib, which is also called Lumakras. We have, and you can see KRAS is in the name of Ludacras and RAS is in the name of sotorasib. So we'll see other drugs with RAS in the name that are RAS inhibitors or KRAS inhibitors. And this is made by Amgen, who really has been pioneering the development of such drugs. Mirati also has a potent KRAS inhibitor called adagrasib that has FDA breakthrough designation and hopefully will get FDA approved soon. But there are a number of other drugs in development for KRAS G12C and others, which we can certainly discuss. So cancers can develop resistance to KRAS inhibitors after initial response. So we know this, and what we know is that the time to development of resistance is variable from one person to another. Cancer can regrow at the sites where you already knew it had been before, or new spots can appear, of course. sometimes we continue the current treatment even though the cancer is growing, and incorporate, say, a local therapy like radiation um, or surgery to a small number of limited sites of growth. We call that oligoprogression. When we say cancer is progressing, it means it's growing. So sometimes we keep the KRAS inhibitor on and treat a single site or two of growth. But how cancer develops resistance, or what we call the mechanism of resistance, really differs between patients and can also differ between one spot and another in the same patient. And so when safe and feasible, we do really try to understand the mechanism of resistance. And how do we do that is with usually a repeat tissue biopsy or blood biopsies. Blood biopsies are also called liquid biopsies that represent circulating tumor DNA. So sometimes the tumors can release DNA into the bloodstream, and that can be picked up by a blood test. So what are the broad categories of resistance that we have been starting to find? So the first category is called on-target resistance, meaning the target that the drugs are working against, in this case KRAS, it was already mutated in the first place, like G12V or G12C, and then after resistance to a drug, another mutation forms in the same target. So, for example, a different spot of KRAS can become mutated. And the reason why that causes resistance is because the drug can no longer bind in the pocket of KRAS where it needs to fit. And we'll talk about a few of these in a little more detail. A second broad category of resistance we call bypass resistance, which means that another gene besides KRAS now becomes abnormal or mutates. So, the drug, is like in this example of sotorasib, is working just fine, but now some other gene becomes a problem and takes over that signal. So an example would be the MET gene, or the RET or ALK or BRAF genes can become abnormal, and it can work its way around KRAS to turn that signal back on. A third example is what we call histology or histologic transformation. That means, what does the cancer tissue look like under the microscope? And in these cases, we've seen that the actual type of lung cancer that we've understood so far changes. So one example is called lung adenocarcinoma, which is the more common type you find with KRAS, and it changes to something called squamous cell carcinoma. And these are different tissue subtypes of lung cancer. This is something right now that we can only detect with a tissue biopsy. We can't find this because we have to look at the cancer under the microscope. We can't find this by a blood test. And then sometimes we really try to, get a repeat biopsy. We send a blood test. We do as much as we can, but we really don't understand the mechanism of resistance. And that's a big category right now that we and others are really trying to kind of elucidate and understand further.
Can we go back a slide? Can we go back a slide real quick? Okay, I just want to make sure I'm understanding so I can use the correct terminology. Okay, yes. So the mechanisms of resistance, right? If my KRAS G12C mutates into another type of the KRAS, then it's an on target.
That's right.
On target,
That's
right?
That's
if
right.
It turns into like something further or another place in the pathway, like BRAF or MET or something, then it's considered a bypass, kind of like an intersection, like traffic, right?
Right.
Okay, and then the histologic transformation is where it completely it kind of morphs
That's
into
right.
a different sort of critter. And what would be included in unknown?
Right, that's a great question. So, in cases in the unknown category, we do a full DNA sequencing where we look again at the KRAS gene sequence, and we don't find any new KRAS mutations. We don't find any abnormal other gene bypassing KRAS. It looks the same under the microscope, and so right now it's still a big question mark for our research community, and it is a big chunk of the pie. And so we're really trying to understand, in cases where the cancer is growing through a drug, why is this happening? And right now, we don't know how this is happening, to be perfectly honest. So that's what our research is really also trying to focus on. So we're trying to overcome and prevent the known mechanisms of resistance, so the number 1, 2, and 3, and also uncover or learn what we don't know so far through research. So it's a really important field. And I'll touch on some examples. I mean, this does really make a difference because there have been examples outside of KRAS where we have been able to develop new drugs to overcome some of these resistance mechanisms. I'll just touch on that briefly. Hopefully that's where we're going to see the KRAS field going soon as well. So again, just to kind of touch on each of these topics further. So here's that same pathway that I showed before. Here's the original KRAS driver mutation or activating mutation. But in the case of on-target resistance, a second spot in KRAS gets mutated as well. And that's where the drug needs to bind to really work. So here is an example of another position further down along the KRAS protein called Y96C. And we had described this as kind of our first understanding of some of the resistance mechanisms. So here's a patient of ours at Dana-Farber who had a KRAS G12C mutant lung cancer. And here's a cross-section of the chest where you can see cancer on both the left and right side. Had a great response to the Mirati drug adagrasib. But then you can see the cancer started to regrow. And at this juncture, we did a new biopsy. And in addition to the original mutation, there was this new acquired mutation called Y96C. And we since have learned that this is really the exact location. And there's a couple other spots of KRAS where drugs like adagrasib and sotorasib really need to fit to work properly. And so potential strategies to overcome this is to think about switching to a different KRAS inhibitor. So a different drug might be able to bind in a little bit of a different way, such that a mutation like this, maybe it blocks adagrasib, but maybe it doesn't block a drug like sotorasib. And I'll touch on some of these nuances, but this is kind of a theoretical strategy of how to overcome this mechanism of resistance by maybe switching out to a different KRAS inhibitor that maybe has a different structure. Here's the other example, bypass resistance. So in this example here, we just have the original KRAS mutation that we started with. So in this example, G12C. But in this example, a different protein like RAF or BRAF became mutated. And here we call it bypass because it kind of works its way around that blockage, the stop sign that sotorasib puts up here on this pathway. And it kind of picks up the signal, picks up the gas pedal and just tells the signal to keep going, tell the cell to dividing. So a potential strategy to overcome this resistance mechanism is, say, to keep on the KRAS inhibitor and add a second drug, like a BRAF inhibitor, for example. Right now, we're just at the cusp of testing these combinations. And sometimes when you start mixing drugs together, they can be more difficult to tolerate. So we need to establish both effective, but also tolerable and safe drugs to combine with one another. And here's an example of that histology change where under the microscope, before starting on the KRAS inhibitor, this was lung adenocarcinoma. And then after resistance to adagrasib, in this example, it changed shape, or it morphed, as you said, Terry, to a squamous cell carcinoma. And this is helpful to know because we can use different chemotherapies, for example, for adenocarcinoma versus squamous cell carcinoma. So we might switch or tailor subsequent treatment based on the histology under the microscope. So is this important to do? Why do we do this? Is there any precedent for studying resistance to fuel drug development? And yes, there is. So in other types of lung cancer, and this applies to other tumor types outside of lung cancer as well, by studying resistance, that has really fueled the development of new drugs and new strategies. And I put a couple examples here. So there are other types of lung cancer that have, for example, EGFR mutations or ALK mutations. And we had earlier generation drugs that would cause resistance. Or that would be very effective, but then resistance would develop. And sometimes, again, here are some on-target resistance mutation examples. So if you treated an EGFR mutant lung cancer with erlotinib, sometimes the EGFR gene would mutate again at position T790M to cause erlotinib not to work anymore, but that led to the development of a new drug called osimertinib or Tagrisso, and that can really overcome this mutation, and now this has become a new standard of care. There's another subset of lung cancer with ALK mutations, and there are older drugs like crizotinib and alectinib and others, but here again, you can develop ALK resistance mutations that can be overcome with new drugs like lorlatinib. So we're trying to work on this understanding for KRAS to see if you develop resistance to one drug, can we develop new drugs or switch out to other drugs to overcome resistance? Here's a couple examples of bypass resistance and how it's important for treatment selection. So on the left here is one of my patients at Dana-Farber who had an EGFR gene mutation that became resistant to Tagrisso or osimertinib. And I biopsied a site of growth of cancer, and there was in addition to the original EGFR gene, there was a new RET gene fusion abnormality. And in this case, I could add a RET inhibitor drug to the EGFR inhibitor, and this peritoneal carcinomatosis, so this patient had a lot of disease in the abdomen causing abdominal pain and digestive issues, really normalized, and he felt much better after starting on the combination. But only by studying resistance were we able to find this new gene had become abnormal to sort of bypass around EGFR. Here's just another example of bypass resistance and how we can use this to develop a new combination strategy. So this was a different patient of mine, also with EGFR mutant lung cancer that developed resistance to Tagrisso, and a repeat biopsy showed abnormality in the MET gene. And so in this case, we added a MET inhibitor pill called capmatinib to the EGFR inhibitor, and this adrenal gland metastasis, which is circled in the yellow here, really shrank down to a more normal size here. So these are examples where only by repeating biopsies and sending blood at the time of resistance could we tailor subsequent therapy as precisely as possible.
Well, let me ask you there, is there a timeline that this sort of thing happens, whether it's EGFR or ALK or KRAS?
Yeah, thanks. It's really variable. So what we are also trying to understand is if you take 100 patients that have the same mutation, like say KRAS G12C or what have you, or EGFR ALK, and treat them all with the same drug, we do see a lot of variation in how long that drug works for. And some patients are still on the drug and haven't developed resistance and don't need to make any changes. And in other cases, patients' cancer regrows more quickly in say 3 months or 6 months. And right now we don't really understand the variation in that way in terms of why some cancers regrow sooner, why some don't regrow, some are very slow to regrow. So the timing as to when these things happen is a bit variable, and we don't have a great kind of way to predict, like if somebody's developing resistance, what type of mechanism might we see based on the time.
So we don't know the timeline, and we don't have the average place that or way that it changes.
Not yet, not with right now we have very limited number of patients where we've studied resistance. So hopefully by studying this in a much larger sense, in a more broad way, we can really understand why resistance happens, when it happens, and in what ways. Just a last brief example is, and then we can go through some of the questions as well in a few minutes, is this histology transformation. So again, this was a EGFR mutant lung cancer where we've had EGFR therapies for many more years than KRAS. So that's why most of my examples are in EGFR, where we've made progress in overcoming resistance. And this is sort of a good roadmap for how we can develop better KRAS targeting therapies as well. Here was an adenocarcinoma that we call a type of non-small cell lung cancer, and here it changed to small cell lung cancer, and here we had to switch to a different type of chemotherapy to treat small cell lung cancer. So these are just a few examples of how since we can't really predict or guess what we're going to find, we really need to interrogate the cancer. We need to understand what's going on, and either by getting a biopsy or sending a blood test. So with KRAS, it's been also complex because sometimes we don't just see one resistance mechanism going on, we see sometimes multiple ones going on. So I have a couple diagrams here where here's 3 different patients. The left side of the diagram represents what we found before treatment or pre-treatment with adagrasib. And the right side represents what we found after resistance to adagrasib. This was with blood testing or the liquid biopsies. And so at baseline, we found the original KRAS G12C mutation, but what we were surprised to see is multiple resistance mutations emerging while on adagrasib. So there were some on-target resistance mutations that are shown here in red, where KRAS mutated again and again, like G12R or Q61 or H95. There were some other gene bypass mechanisms, so a RET fusion or a MEK fusion, which is 2 steps down from KRAS, so BRAF and MEK. Here's another patient example where, again, we were seeing both some on-target and off-target resistance, and again, another patient seeing some on and off-target resistance. Just a few more examples here. So this was a mix of patients with lung and colorectal and appendiceal cancer of the appendix. And again, in many of these cases, we were seeing a few different resistance mechanisms emerging simultaneously. So this also helps us to inform, the principle, can we, switch one drug out for another? Well, it may not be as simple as that if we're seeing a few resistance mechanisms emerging at the same time. Here's an example of a pre-treatment sequencing trace at the top, and after resistance, there's this new spike, which is called MET amplification, as shown here. So we do think there are going to be differences between different inhibitors. And we'll talk about KRAS G12C, but this will apply as we get more inhibitors into the clinic. So shown here is the different drugs, adagrasib in this green color and sotorasib in this yellowish color, bound to KRAS. So this is where the drugs fit into their binding pockets, and this is how they need to work. So they bind to that G12C mutation. That's how they kind of stick into the KRAS pocket. But here you can see there's a few different shapes that are important for the drug to interact with. So R68 and Y96, we found mutations that can cause resistance to adagrasib and sotorasib. This part of the KRAS protein called H95 is more important for adagrasib's functioning than for sotorasib's functioning. So we think, for example, if you mutate H95, it will cause resistance to adagrasib, but not to sotorasib. So we think and anticipate we'll find different patterns of resistance as the different drugs are being tested in clinic. So right now, sotorasib and adagrasib are furthest ahead. But
is H95, I'm sorry, I didn't mean to interrupt.
Please.
Is H95 a different type of a protein, or is it a location?
Yes, thank you, Terry. Sorry, these are different locations along the KRAS protein. So each of these positions here is a different one of those amino acids that forms those beads on a string, and that string kind of folds up into the KRAS protein. So these spirals and spaghetti loops here, these are all amino acids along this string, and KRAS kind of is a protein where these amino acid strings kind of fold together to help the protein to function. So H95 is the 95th position along KRAS, or Y96 is the 96th position. G12 is the 12th amino acid position on KRAS, et cetera.
I still envision like we're playing Battleship, and then it's just where it is, and then based on what that is, and then the letter, what type of a protein it's supposed
That's
to
right.
be.
That's right. Yeah, that's a perfect analogy, right? This is just a diagram here. This was done in the lab of Andy Aguirre, at one of my close colleagues and a wonderful person at Dana-Farber. So here we are testing adagrasib on the left and sotorasib on the right in a model of KRAS with different resistance mutations. So all of them had G12C, and then each line had a different KRAS resistance mutation. And when the drug works well or works better, the cells are killed off. So the line, or this curve, this black line, is all the way to the left. When the drugs don't work, you can try high concentrations of drugs, but the cells are still alive, even at very high concentrations, and those lines are on the right. So with adagrasib, we found in patients, when they developed R68 or H95Y86 resistance mutations, it caused the drug not to work. And some of these mutations also caused sotorasib not to work. So here you can see this R68 and this pink Y96 caused resistance to sotorasib, but theoretically, these H95 resistance mutations could be sensitive to sotorasib. So theoretically, we can envision maybe switching from one drug to another to overcome resistance. But we do need to study this in a much broader scale, and we need to compare resistance to drugs. So this is where we're really hoping to scale up our understanding. That paper that I mentioned, we had published this work in the New England Journal of Medicine last year, was just about 30 cases, and that was a really large effort across many different academic institutions, but we have so many more patients out there, and we're trying to learn from every single patient so that we can advance our drug development and discovery. So this is where we need help spreading the word, or we're looking for participants to understand resistance. So we just activated and launched what we call the SPARK protocol. SPARK stands for Studying Pathways of Resistance in KRAS-Driven Cancers. So right now, this is a nationwide study, so we're unfortunately not recruiting internationally yet. But this is for patients anywhere in the US right now with any type of cancer, like lung, colon, appendix, others, that have a KRAS mutation. And if a patient is on a KRAS targeting therapy, I've only listed a few here, but if the cancer is starting to regrow while on that, that's the patient population that we're trying to identify right now. This is a fully remote participation and remote consent type of study. So you don't need to live near a big city or a big cancer center. You can consent remotely online, or we can help to facilitate that. And after you consent, it's a pretty quick process. We will send you a blood collection kit, and you can either take that to your clinic, or we can also arrange remote phlebotomy to come anywhere that you want, or to your home to draw a blood sample. And all of these costs are covered, so the costs of the blood draw are covered. And this provides a free liquid biopsy to test for ctDNA or circulating tumor DNA to help us understand resistance mechanisms. So this, we're partnering with Foundation Medicine to do the liquid biopsy results, and also working with the Alchemy Foundation, the GoTo Foundation with KRAS Kickers as well, to kind of help in this protocol here. So this is if a patient or if somebody is on a KRAS inhibitor and experiencing disease growth or progression, and they want information about why their cancer might be growing, and we want to help to give that information, because not every oncologist necessarily tests for resistance, or right now there's not per se a new drug approved after resistance develops. So some doctors say, well, why test for it if I don't know, what to do with that information? But we're really trying to understand this now. For patients who aren't on a KRAS inhibitor now, say they were on sotorasib a while ago, if they did have resistance testing done at that time, either a biopsy or a blood test at that time, and they just want to share their data with us, then we have a option as part of the protocol for data sharing, where if you want to send us the clinic note or electronic medical note, we would love to be able to learn from everyone's experience as well. So if somebody has been on a drug like sotorasib, and at the time of growth, if they had to come off the drug because of disease growth, but if they had, say, a liquid biopsy or a tissue biopsy that was explored, please reach out to us, and we would love to be able to learn from that experience as well. So we're trying to learn as much as possible from as many experiences as possible. So how, if people are interested in the protocol, you can email spark at DFCI, which stands for Dana-Farber Cancer Institute.harvard.edu, and we'll get back to you right away. The Dario Lung Cancer Medical Institute site has a link here, but we'll talk also with our collaborative partners about, maybe linking out to some of these, some of our sites here. So if you click on this, you'll go to the Spark protocol page, and it will tell you how to participate. And we really try to get back to people really as quickly as possible. So, so we're excited. We just did a so-called soft launch to make sure everything was working properly, and it's working beautifully. We have a really big team of passionate and dedicated people that are working on this, and we're excited that it's now available for a nationwide launch. So, so
is there a possibility it will be going international as well?
Yeah, we would love to be able to do that as well, Terry. And there are some issues around sort of shipment of blood internationally, and so we're trying to sort that out. And also with this is an IRB, Institutional Review Board approved protocol, but there are some different governing kind of principles around international studies. So we would love to be able to do it. We have some logistics to work out as well that we're trying to sort through. So
okay, and then as far as like if somebody was like on a RAF/MEK inhibitor or like some other pathway sort of a drug, is that work for that?
Yeah, good questions. So we have a number of different strategies now to overcome resistance, and so I'll talk about that as well. So this is a brief list here, I think, apropos to your question, Terry. So what are we trying to do to overcome resistance? So we have new drugs. So of course we have sotorasib, adagrasib, there's JDQ, there's GDC. These are all, so this is made by Novartis, this is made by Genentech, Lilly has a drug, there's many other drugs. These are specific to G12C, but there are a number of so-called pan-RAS inhibitors that are intended to work regardless of what the RAS mutation is. So I know some questions came in about, for example, G13C. So I don't know today that there's a specific trial that's just enrolling G12C, but there are some trials that are enrolling regardless of what the KRAS mutation is. There are studies, to combine KRAS inhibitors with chemotherapy, with immunotherapy, like pembrolizumab, which is Keytruda, or nivolumab, which is Opdivo, but this may be too toxic to advance safely into the clinic. So there have been a few small trials out there already where, unfortunately, in a limited experience so far, there were some cases of liver inflammation that were more on the serious side. So, and Terry, as you asked, there are other trials aimed at combining KRAS inhibitor pills with other therapies, for example, RAF or MEK inhibitors. There's a company called Veristem that makes VS6766. That's a so-called dual inhibitor of RAF and MEK. Or there's also SHP2 inhibitors, SOS1 inhibitors. These are different parts of that pathway in the diagram earlier. And then there's also EGFR antibodies like cetuximab or ramucirumab that have been combined, and many other trials that are out there. So we are hopeful that new combinations will either delay resistance or prevent resistance from emerging when used up front, or if a patient has been on a single drug like sotorasib, for example, we hope that some of these strategies will be able to overcome resistance. Many of these studies are still in early phase, and in early phase, like a phase 1 trial, one of the main goals is also to make sure that it's safe to combine these drugs together. And unfortunately, we've been learning that in some cases, even if it seems, maybe like a promising strategy, it's just not safe to use in a larger number of patients. In other cases, we've been able to combine drugs safely, but have seen so far, not much activity. So I think we really need to advance these efforts as quickly as possible. And I think what we're hoping to do ideally is, I don't know that there's going to be a one-size-fits-all strategy for overcoming resistance. And so ideally, we can inform subsequent treatments by understanding resistance. So for example, if there is MET amplification or EGFR signaling, we could use strategies to target those abnormal proteins. Or if there's a new BRAF or MEK mutation, we could use BRAF or MEK inhibitors. But right now, I think because our understanding of resistance is limited, we haven't been able to, for example, have a patient where the cancer is growing on a drug like sotorasib, get a repeat biopsy or a blood sample to study resistance, and then if there's a specific mutation, then direct the patient onto that specific kind of targeted therapy as the next step. So right now, where some of the trials are, it's just taking patients whose cancer has grown on, say, sotorasib, and give them all the same therapy. But we really need to kind of understand this better to try to personalize therapies as much as possible, both up front, but also if resistance develops. Again, here's just an example of some of the SHP2 and SOS1 inhibitors that are in development, and I know you've been working closely with, and I'm monitoring the chat here, but we can also go through many of these questions too. So, companies like Novartis, Revolution, Boehringer are working on many of these inhibitors as well. There's work and trials will be opening for KRAS G12D. So, for example, Mirati, MRTX1133. This is all what we call preclinical information. So before it gets into the clinic, we're seeing some cell lines, so cells grown in a petri dish or in an animal, and we're seeing some impressive tumor shrinkage with this drug. So hopefully this drug will be safe and effective for G12D. We have also Astellas has a G12D degrader, and so many new therapies about to open or already starting to open in clinical trials. We've got the Revolution Medicine therapy that is intended to work across different tumor types. So this CRC is colorectal cancer, and PDAC is pancreatic ductal adenocarcinoma, so pancreatic cancer. And here are some G12D examples where the control is being tested against different amounts of different doses of the drug in a mouse. And here you can see the control where they're not getting the drug, the tumor just keeps growing. And then at the two different doses here, you can see that the tumor doesn't grow at all. So we call these xenograft experiments. And again, we're starting to see, these are all preclinical models of pancreatic G12V and another model of colorectal G12V where you don't see the tumor grow, and in fact, it stays the same, or in this case, shrinks here. So we're really excited to get these drugs into clinics across the world to learn how well they work in different types of KRAS lung cancer, and of course, learn about their safety and whether they can be combined with other treatments. Again, these are different models of lung cancer here, pancreatic cancer, colorectal cancer. So anytime a bar goes down, that means the tumor is shrinking. If a bar goes up, that means it kept growing. So the response rate, ORR, is really what percent of the bars are going down. So you can see here, again, these are all animal studies, or what we call in vivo preclinical studies. But again, hopefully these laboratory findings will translate to clinical activity or tumor shrinkage for these different tumor types. And here at the bottom, you can see what type of RAS cancer these models had, and some were G12C or D or R or S or B. So again, we're hoping to really expand upon our understanding of RAS inhibitors and how they can work and then work in different tumor types. So I want to leave time for questions and going through the chat, but just to say, we of course see these KRAS mutations in many types of cancer. There's thankfully a growing number of options to treat these cancers. We know that cancers can develop resistance, and we do think it will be helpful to characterize resistance through repeat biopsies and blood tests. Hopefully the SPARK protocol will be a good mechanism for us to learn as much as we can, as quickly as we can from every patient, and not, for example, just focus on patients that I'm seeing just at Dana-Farber. We want to study as much as we can to advance the field. And hopefully by understanding resistance, that will fuel our drug development to delay and overcome resistance. So with that, I will thank you all for your attention, and I want to open it up for more discussion.
Okay. Well, great. Well, thank you so much for bringing that all in. And if you want to put in the chat the link to the SPARK protocol, that'd be great.
Perfect.
But okay, so let me make sure I review so that I understand if I need to know. If you've ever been on any sort of an inhibitor, whether it's adagrasib, sotorasib, GDQ, JDQ, whatever, okay, any one of the inhibitors, okay, then, and I would be able to participate in this in the US.
That's right. That's right. So, and ideally, so if you're on one now currently, or about to go on one, and if there's a point, hopefully not, but in the future where your oncologist is saying, or the scans are showing some growth, if you are able to reach out, we can provide the kit, within a couple of days and ship it to you right away. If you were on one in the past and had to come off a drug, but and switch to other therapies, if at the time you were the cancer was growing, or you were coming off therapy, if you had a repeat biopsy or a blood test, a liquid biopsy that time, and just want to share that data or information with us, please feel free to reach out. If your doctor didn't do testing at that time, and you've moved on to other therapies or chemotherapy or something not quite targeted, that can sometimes be a little bit harder to interpret the resistance mechanism if you're currently on other treatments. But we field many questions, and so if you have any question or query or interest, just reach out, and we would be happy to kind of talk through whether you would be eligible now or maybe eligible down the road. So yeah, in the chat, I put our email. I also put a link to the Alchemy website that describes the SPARK protocol, and we can, our team can help walk anyone through questions. Yeah.
So if in doubt, ask.
Yes. Yeah.
Okay, all right. Well, we're going to assume that people qualify unless you tell them otherwise, if they've been on some sort of an inhibitor, and they can just kind of go directly to that. That'd probably be the fastest way of answering. So, okay, I'm going to scroll back through the chat and see. Before we go jumping back to the chat, does anybody have any questions specifically around the SPARK protocol before I go back through the chat, and because I'm sure some of the things might have changed? I'm not seeing, if you see anything, just give them a holler out. Can a patient participate in SPARK if resistance to sotorasib occurred a number of months ago?
Yeah, feel free to reach out. I think if we would kind of love to hear more details. I think if you had like a biopsy then, or a blood test then certainly, or if even you just want to share your data with us, you'd be welcome to do that. If you came off sotorasib several months ago and went on to other therapies, we are right now not doing the kind of blood testing at that interval because other treatments hopefully will kind of modify or change what we can find in the blood. So we're really trying to capture resistance right as it's developing. But
this is where the whole the biology combines with information, right? IT stuff is we're just building up a big data warehouse so you can see all the information.
That's right.
As far as and look for trends and correlations to see
That's
if
right.
you can find any similarities. If people have
Yes.
this plus this, and I don't know, the brown-eyed people are more inclined to get this resistance.
That's right.
Whatever it may be.
And right now, the only way we can do it is, I'll reach out to colleagues at other cancer centers or institutions and say, hey, can we, try to combine experiences together? And at most, we've been able to get, dozens or 100 patients or so, which is a lot based on what we know so far. But I think the hope is that, we can drive the research forward with patient participation everywhere. And there's thousands of patients out there. And if we can really scale up, I think that will really enhance our understanding.
Okay. All right. Fantastic. Well, I'm excited to be able to share that information with the community, and hopefully the more information, the more power we've got towards it. So I'm going to circle back to the top of the chat, and I'm going to kind of work my way down.
Yeah.
And so we'll probably be jumping around conversation topics a little bit. Says, hi, Dr. Awad, for KRAS G12C inhibitors trials, can one start with only one KRAS G12C inhibitor and in case of resistance move to another combo arm? Let's see. Or does it make more sense to start with the combo arm?
It's a great question. Every trial is a little bit different in terms of moving between arms. So every clinical trial has a unique set of what we call eligibility criteria or inclusion and exclusion criteria. And I, of course, love the questions about clinical trials because that's really how we advance our understanding and get new drugs into, approved. So I love, I know it's not easy to participate in trials, and we ask a lot of our patients and their families to participate in trials, but hopefully it will really make a big difference. So some trials say if you've been on a prior G12C inhibitor or a specific type of KRAS inhibitor, you are still eligible for this trial. Others will exclude some prior therapies. Every trial is very finicky in a way, and sometimes they'll say, you cannot have had more than 3 different lines of therapy or 3 different types of treatment for this type of cancer. If you've had more than that, you're not eligible. If you've had fewer than that, you're eligible. Some trials have multiple arms where they're exploring different combinations, and they may allow a patient to switch if they tried one arm and then switch into another arm, but not every trial is like that. So it's a little bit tricky. We don't have data yet to know that a specific combination upfront is better than starting with a single drug by itself. That's what we really hope to learn in trials. Theoretically, the principle behind a combination study up front is you're hitting the cancer from different angles or different directions, which would hopefully delay or prevent some resistance from emerging. The unknown is, if it doesn't work better than the single drug by itself, are you unnecessarily exposing somebody to the potential side effects of mixing or adding drugs together? So I think there's theoretical, pros and cons to trying a single drug by itself or a combination. But I think it's a good idea to talk to your doctor about, in the case of a specific combination, how much is known about the other drug that, say, sotorasib is being combined with. Some drugs are further along, and they're already approved, or we know about them from years of experience. and other drugs are in phase 1, where only handfuls of patients have taken them in the world. And so I think when considering combination studies, talk to your doctor about what's known about the other drug. I think drugs like, I see a question mentioned Avastin or bevacizumab. I think we don't have a lot of data yet in KRAS as to whether combining an inhibitor with Avastin is better than the oral inhibitor alone. So, but it's always good to try to explore these questions.
Wasn't something recently fast-tracked with, there was a Bridge Bio drug. I don't recall what kind of a drug it was. Was it SHP2 inhibitor or something? Let's see.
Yes.
Yes. I don't remember what the name of the drug was. But I could have sworn I saw that very recently. And it was in conjunction with the sotorasib specifically.
Yeah. Might be.
You know what I'm talking about, or am I just did I have some weird dream?
No, thank you. I think BBP is the Bridge Bio drugs, and I think 398 was their SHP2 inhibitor, and then
I think that's what it was.
Yeah, and then 454 is a KRAS.
I don't remember the number. I just remember
Yes,
the
it's hard
Bio
to
drug.
Yeah. Yes.
And how does that fall into the scope here?
Right. So so KRAS has to propagates that signal by interacting with other proteins so SHP2 is one such cooperating protein, or SOS1 is another such protein. And in the lab, it does seem like combining a SHP2 inhibitor with a KRAS inhibitor or a SOS1 inhibitor in the lab on cells in a dish, seems to be more effective than using the KRAS inhibitor alone. Now, there's been a lot of interest in these, and some trials are further ahead. I haven't seen data on that Bridge Bio compound recently yet, Terry, from patient experiences. But one of the challenges has been with some of the SHP2 combination trials is side effects without an apparent benefit over the KRAS inhibitor alone. And so I think it would be great to keep exploring, and maybe there are different ways of dosing the SHP2 inhibitors. Maybe you don't have to take it every day. Maybe intermittent dosing would be more effective, but safe. But right now we don't have convincing data yet about any particular small molecule combination. But hopefully, as these studies mature, we will start to learn more about what strategies might work or might not work.
Is there been any difference in the colorectal versus pancreatic or lung and resistance with the inhibitors?
Yeah, it's a really, really interesting question. And this is something that we want to study in a broad scale. So in our original paper, and Sloan Kettering had since also put out a nice paper on resistance. Again, our numbers were small, but we were getting the sense that in colorectal cancer, resistance, there may be more of a propensity to develop multiple mutations simultaneously, or what we call acquired gene fusions. So I don't know that for sure, that these were really small numbers, so hard to kind of know from a kind of statistical standpoint if there was a meaningful difference. But that's something we want to do by studying hundreds and hundreds of patients if we have a good cohort and understanding of resistance to drugs in lung cancer or colorectal cancer or other cancers. I think we'll be able to start seeing patterns emerge that explain differences across different tumor types. And if we can look at everybody treated with, say, sotorasib versus adagrasib versus Revolution Medicine or others, hopefully, again, we'll get a better sense of resistance mechanisms.
Will there be any sort of, when you're pulling that data, will you be looking at the preliminary testing as well? Meaning like whether there's co-mutations of, STK11 or something as well?
Yes. Yeah, perfect. Thanks, Terry. And we're working on this with a multi-institutional collaboration right now also that, but we want to study this with the SPARK protocol, because we're asking if patients have access to the original genomic report that found the KRAS mutation in the first place. We would love to be able to see that as well, to know, what else was going on in the cancer besides KRAS, and there can be what are called co-mutations or other mutations that coincide or co-occur with KRAS that may help us understand variation in response or duration of response from one person to another. So yes, we would love to look at baseline factors as well as acquired factors that give us a sense of why the therapy is working out.
Yeah,
well.
your goals are probably lower than mine, truthfully, because I'm thinking, all right, I know a lot of people that had a lung cancer, might have been like a stage 1, stage 2, and then there was no genomic testing done because it wasn't a thing. And then years later, they had testing, it might have been like a G12C, and they're able to like pull that in the meantime. They may or may not be on an inhibitor, and kind of like watch it as it kind of comes down the pipe.
Right. I think that would be fantastic also to kind of follow that prospectively.
Yeah, I hope so. Okay, well, I'm going to keep cruising through the chat. Have you had any experience with G13C and any trials related to that?
Great. So G13C, and I'll mention, I think another question came, about using drugs outside of G12C. So drugs like sotorasib and adagrasib and JDQ and GDC 6036 and others, they are very specific to G12C, and they don't work on G12V or G12D or A. And the reason for that is because they form a bond specifically with the mutated amino acid. So when that glycine amino acid at position number 12 changes to cysteine, these drugs bind to that cysteine exactly at that position. But if there's a different amino acid there, like G12V or the drug doesn't bind at all, and they don't work at all in non-G12C mutant cancers. And so I think there's not as much known. G13 mutations are recurrent in cancers, and they're not maybe as common as G12C, but really important to identify, because I think hopefully some of the pan-RAS inhibitors or RAS degraders may work in G13 or Q61 types of RAS mutations. So hopefully we'll see some.
Okay, well, why don't you check out and see if this is my good explanation. Okay, so you can be my fact checker for me. All right, so I think of it like if the protein my body was supposed to be making a G on my 12, and instead made the C. That is like me taking my front door and putting it at my neighbor's house. It doesn't mean I moved. It means they have my front door, and it's the wrong door for their house. Is that
That's
a good
right.
explanation?
Yeah, I think that is. Yes. Right.
Okay, or at least accurate. How about that?
Right,
But accurate.
Yes, right.
Okay, all right. Okay, I'm going to keep cruising down. Current incidence of resistance to sotorasib, is there an average time? I think you kind of said no, was kind of what the short answer was, right?
Yeah, I think in clinical trials, you might have heard of this term PFS, or median PFS, progression-free survival. And so what does that mean is what's the average or median length of time without progression. So if you take a bunch of patients with KRAS G12C and they all get sotorasib, on average, or about half of patients might have their cancer progress. The number is between like 6 and 7 months. So is the median PFS on some of the sotorasib trials. So half of patients' cancer might start to regrow within 6 months, and the other half of patients, don't have growth within that time, and may, if they experience cancer growth or progression, it might happen after that time. So it's a meaningful step forward for KRAS. We want to do much better. Some of our other inhibitors or drugs have longer median PFS numbers. So on average, those are sort of some numbers about when resistance may develop or when cancer might start to regrow for patients.
Next question, are the on-target resistance mechanisms occurring in with the original KRAS mutation? I'm not in conjunction, I'm not sure what.
Cis, okay,
Yeah.
right. So we've seen usually the cancer will still have the original G12C mutation and then acquire additional KRAS mutations. And somebody asked David Driscoll if they occur in cis or the other thing is called in trans. So what does that mean is, we all inherit two copies of KRAS. One is from the maternal side and one is the paternal gene. And usually the original KRAS mutation is just on one of those copies or one of those genes. And so the two options are a new KRAS mutation develops on that same gene that the maternal side, say, for example, and that we call in cis, or it's sort of in together with the original mutation is the new resistance mutation. And then the other option is that in trans or on the opposite gene, from say the paternal side, a new KRAS mutation develops. And we have seen both occur, some resistance mutations in cis and some in trans.
Okay. And what is percent of those that form resistance and those that don't? Even if it's just kind of like it's trending this direction.
Yeah, I think we found a sort of a resistance mechanism in our cohort, maybe about say 40% of the time, 30-40% of the time, we could kind of say, okay, I think this probably explains why the drug wasn't working as well anymore. But then there's that large portion of the pie chart of unknown resistance mechanisms that we're really trying to do other analyses of the kind of RNA or the what we call epigenetic or non-genetic mechanisms of resistance. So we're really trying to understand how this is happening.
So when you say unknown, you just mean that there's some sort of resistance we have not identified exactly, like it's mutated into something else. It's just, it still needs further review. It's just too murky.
Right. I think, our publication on resistance to G12C inhibitors was just a year ago, and that was the first description of resistance. And so there, I think we're really scratching the surface, or it's the tip of the kind of iceberg of understanding.
Okay. So do you expect a big difference between different G12C inhibitors efficacy, or do you think main difference is what the best combo is of the G12C?
Right. It's a good question. One question that our community is asking is, are we hitting a kind of therapeutic ceiling with the different drugs, or can we find a drug that, is better than the rest in the crowd? So I think right now we have the most data on sotorasib and adagrasib, and I think they seem generally comparable in terms of their efficacy. They might have a little bit different side effect profile, but the data that have been shared so far, presented publicly so far from Novartis on their JDQ drug 443, or Genentech's GDC 6036, or Lilly, or it's very small numbers of patients, so it's really hard to know if in a larger cohort of patients if the response rate will be much different. So I want to be, I'm always an optimist, and I want to, hopefully see one of these drugs really stand head and shoulders above the rest. But we may need to kind of keep moving past and think about the best combinations with these drugs by themselves.
Okay. Do you expect appendix cancer with the G12C mutation to behave the same or similar to CRC with the G12C?
Good question. I think appendiceal cancer, we had one in our initial New England Journal paper about resistance. And so, even though it's an uncommon or less common cancer type, we really want to study that as well. So we only had one example in our case series of appendiceal cancer. So I anticipate it may behave similarly as colorectal cancer, but I think it's hard to know.
Okay, if somebody had appendiceal cancer, it's mean there's not necessarily a trial for that. Can you be included in like a CRC arm?
Some trials have what are called tumor agnostic or arms, where as long as there's a G12C mutation, and if you've tried kind of a standard treatment already, then you could enroll to such a study. So some protocols are specific. So if they say colorectal cancer, they may specify if they'll allow appendiceal or not. So it's a little bit specific to each trial.
Okay. Can you go from one inhibitor to another, or do you need
Right.
something else between?
Good question. Right now, we don't have any case reports or examples of a patient. There are trials that are looking at this question. If you were on sotorasib and switched to adagrasib or vice versa, or go on to JDQ or GDC, so we don't know if it will work. I think ideally, if we know how the cancer became resistant to the last inhibitor, we hope we can use that information to kind of guess or predict whether you would be sensitive to switching to another drug. So for example, theoretically, if you are on adagrasib and had that H95 resistance mutation as the only mechanism, then I would guess, the cancer could respond to sotorasib. But we don't have examples yet in the literature or in my clinic experience of seeing, swapping out a different inhibitor, rescuing, what happened after the last drug. So depending on what you've been on before, sometimes we do switch classes of medicines to other chemotherapies, immunotherapy, antibody drug conjugates, or other such treatments.
Okay. Resistance. Have you seen any resistance for appendix cancer so far?
In our case series, we did have that one case of appendiceal cancer, and it did have some what we call acquired gene fusions. It had, for example, it had a mutation in a gene, called NF1, for example. So it was just one such case. But again, hopefully with a broader experience, we'll be able to understand what may be specific or unique to appendiceal cancer.
Okay. I'm going to say if you have a question about the SPARK trial, please reach out to them. What trials, drugs are in the pipeline for general KRAS mutations? G12C is not very common for colon, and so other options are needed.
Yes. So I know I kind of flew through some rapid examples towards the end, but there are these so-called, pan-RAS inhibitors that are focused on KRAS mutant cancers, regardless of the type. So, for example, if there's G12D, for example, there are some G12D specific trials. There's the Revolution Medicine and other such drugs that are so-called pan-RAS inhibitors that may work against G12V or D or S or others. So, for example, RMC6236 is that Revolution Medicine compound. So I think there are hopefully some new agents that are on the horizon for different types of KRAS mutations.
Okay. Although the combo of KRAS inhibitor and immunotherapy may show dangerous adverse events, we heard some info regarding these two therapies alternating one after the other, but not at the same time. Would this be safer?
Right. So there were some data presented about, for example, sotorasib in combination with pembrolizumab, which is Keytruda, or sotorasib in combination with atezolizumab, which is also called Tecentriq. And that's where some higher grade liver inflammation or hepatitis toxicities were first reported. They're trying some, maybe lowering the dose of sotorasib, or adagrasib is also trying these combinations, or doing a bit of a staggered start where you start with so-called lead in of one drug, and as long as you're tolerating it, then add the second drug, and then continue both. Alternating is what we do now in clinical practice. So, for example, in lung cancer, the G12C inhibitor drugs are approved not in the first line setting, but in the second line setting after you've been on something else. And so there we do switch from one type of drug to another. So you might be on immunotherapy or chemotherapy plus immunotherapy and then switch to a KRAS inhibitor. The one thing that we're trying to understand is some of the immune therapy antibody drugs like Keytruda or Opdivo and other, the medicines have a long half-life, meaning they stay in the body for a long time. And so even though you are, switching off of immunotherapy and stopping it in order to start a KRAS inhibitor, we are trying to understand the safety of that. In other words, is there still kind of some level of that immunotherapy drug in your system, even though you're not continuing to receive it, and could that predispose to some higher rate of side effects than you might have otherwise had, without prior immunotherapy. So that's one question we're trying to address through some of our analyses. The KRAS inhibitors, the pills typically have short half-lives, meaning they get out of your system more quickly. So going in the opposite direction, so instead of immunotherapy and then a KRAS inhibitor, because the immunotherapy might stick around in your system for much longer and interact with the KRAS inhibitor, it may be safer to sequence things in other ways, just from a safety standpoint. Now, that might not be the most effective thing for the cancer. So that's something we try to take into account, when meeting a patient, is what's the best therapy to offer up front. And, if that doesn't work and we need to switch to something else, what's the best and safest therapy to offer next? So I think we're trying to learn what is the best sequence of therapies, what's the safest sequence of therapies. So there's not a per se right or wrong answer, but that's something we're trying to learn.
So identifying the strategy for each individual person based on their situation.
Right. Sometimes we'll use things like the PD-L1 level if it's very high or low, or if there's also a STK11 or a KEAP1 mutation in lung cancer or others. So, we try to take into account, a lot of factors to choose the best therapy.
Okay. Let's see, current incidence resistance to sotorasib, average time. You kind of like, did you answer that? You kind of like,
Yeah, the I guess average time about resistance to sotorasib. That when we were talking about the median PFS is about like 6 months, so that's sort of on average when most what half of patients will see their cancer start to regrow on sotorasib. So in terms of so some people have to switch off of sotorasib within 6 months, about half of them do, and then others have to can continue on beyond 6 months. So that's kind of the average sort of time period, but for resistance to occur with sotorasib.
okay. Any news on the Revolution Medicine?
We, for example, at Dana-Farber are working to open their trials as imminently as possible. So I haven't seen any preliminary clinical data yet, but hopefully.
Okay. Any estimation when that might be?
I think we are hoping to open it. I think it's we have these few steps that we have to get through, which we always want to open our trials sooner than the process.
We want you to open them sooner too, but
I
we
know.
know.
So I anticipate within 1 to 2 months, I hope.
Okay. So coming soon.
Yes.
Okay. All right. Here, I'm going to say this directly. If an oncologist told you to try a G12C inhibitor for a G12D change oncologist, see a specialist. That's my personal take on that. Would that be accurate?
Yeah, I saw that question. Right. And so, these are new drugs, and I focus on lung cancer and some other very specific situations. But for me, I try to stay abreast of developments in all tumor types. But like if somebody had colorectal cancer, I would want them to see one of my colleagues that focuses on colorectal cancer, just because there's so many nuances, which is good, that if you can see a specialist, or intermittently see a specialist, or if you're at a juncture where your oncologist is saying we might need to switch therapy to this or that, if you have time to kind of check in or do telemedicine with a specialist, I think that's a great option, because I think if they recommended sotorasib for G12D, it does reflect maybe, that oncologists might have a hard time kind of keeping up with the new information, or they might not understand how this drug works, that it really only works for G12C. I think trying it for G12D would unfortunately just expose somebody to side effects, and not be effective at all. So I think it would cost time and potential side effects.
Yeah. Okay. Info gathered by the Spark help getting some of the combo KRAS inhibitors plus other things approved more quickly to give access to patients who are running out of options. That's a good question. It was the last question in here, and then.
Thank you. Yes. We hope so. I see the question here. So, what we've been pleased to see. So after we published our initial report on resistance, all the other companies now have to show whether their drug can overcome those resistance mutations that we reported on. So I've been happy to see that. So we published on H95 and Y96 and R68 mutations. These are the different positions in KRAS. And so now when I talk to Novartis or Lilly or Genentech, and they're trying to kind of explain or articulate, is their drug different or similar to the ones that we already have, they need to show these data too. So we hope that by characterizing resistance through the Spark protocol, that will really fuel drug development and drug combinations to hopefully overcome resistance. So I think I would love for our work to really help accelerate discovery and drug development, and for the hope of improving outcomes for patients on trials or elsewhere. So yes, I hope through our learning and understanding, through all of our patients and advocates out there helping and participating, I hope we can all kind of crowdsource the information as much as quickly as possible to advance our fields.
Yeah, and it seems like the data has a tendency to drop very heavily in the spring. And I'm not sure if that's just the timing of the conferences or the fact that, the holidays, right? I mean, we're almost on top of it.
That's right. Yeah.
Yeah. So yeah, it's probably when I'd be looking for some of that data on RepMed. Let's see, I think I missed something here too. Data for colorectal about combination of G12C inhibitor with immunotherapy. Did I ask that already? I don't think
I haven't.
And I know you're a lung doc.
No, that's okay. I stay abreast because I collaborate closely with my gastrointestinal, colorectal colleagues. That Andy Aguirre, that doctor I mentioned earlier, is a GI oncologist. So we're working really closely together to understand differences between tumor types and response to these therapies. I think I haven't seen as much data yet in colorectal cancer, but the combination of G12C plus immunotherapy with sotorasib, I think, has been toxic, or there have been a lot of adverse events regardless of the tumor type. So, we'll see if other drugs from Mirati, Novartis, Genentech, others can be safely combined with immunotherapy. I think we just don't have that information yet.
Okay, well, I mean, I want to be respectful of your time. And I don't think I missed anything other than thank you for being amazing and wonderful. And once again, the amazing Dr. Awad and all this. So,
We should have a whole nother session just for the.
yeah,
those-
we'll have to spend a couple hours telling you how amazing you
Right,
are and how
right,
wonderful you are and what have you. So,
Yeah.
you know, we always learn so much from having you here and your explanations and what have you. And so I appreciate taking the time to do this and, you know, share with us what's going on. I'm very, very excited about the Spark protocol.
Thank you
And,
so much.
you know, the more we hold that data together, the better opportunity we've got for learning.
Thank you, Terry. I always learn from you and from the KRAS Kickers and try to learn from every patient to help them and to help patients who are coming after them as well. So thank you so
Yeah,
much.
so thank you so much. And we're going to record this. And we'll put this on. And I hope you have a wonderful Thanksgiving and holiday season.
Thank you so much. Pleasure.
Okay, thanks for joining.
See you soon. Thanks, Terry.
Okay, take care. All right, thank you all. Okay, well, we're logging off now.